Hello everybody, my name is Nobonita Rai Chattopadhyay and I am working as an assistant professor in the Department of Biotechnology, Haldia Institute of Technology. Today I am going to tell you a few things about bioprocess engineering and I will discuss the chromatographic techniques in greater detail. Bioprocess engineering is a stream of engineering sciences where you can generate marketable products by the use of biological living cells or biological entities. It can broadly be divided in two parts. One is called upstream bioprocess and the other part is called downstream bioprocess. In upstream bioprocess, we can follow a few steps to select, modify, enhance, and develop the living entity which can be used for making the marketable product. After generation of the product, we have to purify that before marketing it. In downstream bioprocess, we follow all the steps by which we can purify our desired product and make them ready for the market. Usually, the purification part of the downstream bioprocess engineering involves various types of chromatographic techniques. So today I will discuss various types of chromatography. The term chromatography is derived from the Greek words chroma means color and graphein which means to write. The method was developed by the Russian botanist Mikhail Tsvet. He developed the technique and coined the term chromatography. He used it primarily for the separation of plant pigments such as chlorophyll, carotenes, and xanthophylls. Since then, different types of chromatographic techniques have been developed which are useful for many separation processes. These processes are so important that more than 12 Nobel Prizes have been awarded till date. Chromatography is a technique for separating the components or solutes of a mixture. It is done on the basis of relative amounts of each solute distributed between a moving fluid stream, that is called a mobile phase, and a non-moving stationary phase. The mobile phase may be either a liquid or a gas, while the stationary phase, which is non-moving, is either a solid or a liquid. The mechanism of separation depends on the kinetic molecular motion of the solutes present in the mixture. Kinetic molecular motion continuously exchanges solute molecules between the two phases. For a given molecular species, the ratio of the time spent in the moving phase and in the stationary phase is equal to the ratio of its concentrations in these two phases, known as the partition coefficient. During operation, a mixture of the solutes is introduced into the system in a confined region or a narrow zone. Usually that is called the origin or the line of sample addition or the spotting line, whereupon the different species are transported at different rates in the direction of fluid flow. The driving force for solute migration is the moving fluid, that is the mobile phase, and the resistive force is the solute affinity for the non-moving phase, that is the stationary phase. The combination of these two forces as manipulated by the analyst can produce the separation. Now, chromatographic techniques can broadly be divided in three classes. The first one is thin layer chromatography or TLC. Here, the stationary phase is a thin layer of chemicals supported on glass or plastic or aluminum plates. The next category is paper chromatography or PC. Here, the stationary phase is a thin film of liquid supported on an inert support like paper. And the third category is column chromatography. Here, the stationary phase is packed into a glass column. It can again be divided in 5 major categories: adsorption chromatography, hydrophobic interaction chromatography, ion exchange chromatography, gel filtration chromatography, and affinity chromatography. Now I will discuss all these one by one. Thin layer chromatography. As I have told, a thin layer of chemicals are supported on glass plate. Here in the image you can see that the TLC plate is put inside a chamber which is called the TLC chamber. The mobile phase is a liquid here. You can also see the spotting line for addition of samples, and then as the mobile phase will move through the stationary phase, it will carry the solutes with it And depending upon the partition coefficient, different solutes present in each sample will move apart from each other, and finally we can get colored spots in the TLC plate. The retention factor value for each spot should be calculated, and it is characteristic for any given compound on the same stationary phase and the same mobile phase for the development of plates. Therefore, for a set of stationary phase and mobile phase, different solutes will have different RF values, and they can be compared with each other to finally determine the type of the solute present in the final TLC plate. Here you can see how we can determine the RF value for each solute. Now is paper chromatography. Here, the partition chromatography uses thin filter strips as carrier or inert support. The factor governing the separation of mixtures of solutes on filter paper is the partition between these two immiscible phases. Of those two immiscible phases, one is usually water adsorbed on cellulose fibers, which is present in the paper strip, and that acts as the stationary phase. The mobile phase, that is the second immiscible phase, is the organic solvent which flows past the sample on the paper and generate the solvent front. Now, partition occurs between the mobile phase and the stationary aqueous phase bound by cellulose. The isolation of each solute depends on partition coefficient of that solute. Again, partition coefficient can easily be calculated by the following formula. Like K is equal to concentration of the solute in the stationary phase divided by the concentration of the solute in the mobile phase. Here also you have to remember that the K value or the partition coefficient value is fixed for a particular solute in a particular set of stationary and mobile phases. This is the process of paper chromatography. In the left hand side, you can see the scheme of the technique, and in the right hand side, you can see the original image of a paper chromatography final plate. Next comes column chromatography. As I have told, the stationary phase is packed into a column, and the mobile phase is a moving liquid or a gas. According to the mechanism of separation of the solutes, 5 major types of column chromatography are seen. They are adsorption chromatography, gel filtration chromatography, hydrophobic interaction chromatography, ion exchange chromatography, and affinity chromatography. As I have just told, the stationary phase in column chromatography is packed in a narrow tube which is called the column. After packaging of the stationary phase, the mobile phase is forced under pressure or under the effect of gravity, and then it will percolate down through the stationary phase. When the void spaces of stationary phase is completely filled with the mobile phase, then the flow of the mobile phase is stopped for a few minutes, and then sample is layered carefully on the top of the mobile phase. And then the separation is allowed. Separation occurs under pressure or under the effect of gravity. For active separation, the mobile phase must be poured down after layering the sample, and eventually we can get different fractions for different solutes as shown in the lower part of the image in right hand side. The basic steps can be summarized like this. The column will be packed with cotton or glass wool to block the stationary phase from getting flushed down the column. Then we have to prepare silica or alumina slurry and let the particles to become activated, and we will pour down the slurry in the column. We will allow that to settle and drain the liquid and start running the mobile phase, and then sample layering, and then again mobile phase, and finally we have to flush the column with the mobile phase to remove any residual chemical which may have stayed within the column. Here are a few definitions which are important for column chromatography, like what is the solvent, what is the developer, what is effluent, what is eluent, what is eluate, and what is retention volume. Solvent is the mobile liquid phase with no affinity to the stationary phase. The developer is any liquid which is needed for more affinity towards the stationary phase than the solvent, but less than the solutes, and just capable to move them through the column. This thing can again be considered as a type of mobile phase. Effluent. Any liquid that passes out of the column is called an effluent. An eluent is any liquid that has lesser affinity to the stationary phase than solutes, but is capable of moving down the solutes out of the column. So it is used to elute the solute from the column. Elute is the fraction of the eluent containing a required specific substance, that is the desired solute from the mixture. Retention volume or retardation volume is the volume of the mobile phase that passes out of the column before any kind of elution of any specific solute starts. After separation, the elution of the separated molecule can be done by two broad techniques. One is called isocratic elution. Here, the addition of a solvent mixture of fixed composition during the whole process is performed. In the next type of elution technique, which is also called as gradient elution, the solvent mixture used for elution has no fixed composition. In one variety of gradient elution, continuous change in the composition of the mobile phase is used, and it helps in eluting different types of solutes which is separated inside the column. In the next type of gradient elution, a sudden change in the composition of the mobile phase is used, followed by a period where the mobile phase is held constant. By this type of elution techniques, we can elute out the desired solutes and can use those for further processes. Now the types of column chromatography. First is adsorption chromatography. Adsorption is different from adsorption. Adsorbents are such molecules which can attract few other molecules just on the surface of themselves. The most common adsorbents are alumina and silica gel, in which the interactions with the solute molecule is due to OH groups present on their surface. So obviously more polar molecules are adsorbed more strongly and thus will elute more slowly. Strength of adsorption of polar groups on polar support is in the following order. If the mixture of sample contains olefins, ethers, esters, lactones, aldehydes, amines, phenols, acids, everything, then olefins will come out first and then will come ether, then ester, then lactone, then aldehyde, and finally at the end we will get acids. Adsorption chromatography can be used in separation of various natural products. Like alumina can be used for getting sterols, dyestuffs, vitamins, esters, etc. Silica gel can be used for separating sterols and amino acids. Carbon can be used for peptides, carbohydrates, or amino acids. Calcium carbonate can be used for separating carotenoids and xanthophylls, and so on. Next is gel filtration chromatography. This type of chromatography is also known as size exclusion chromatography or molecular exclusion chromatography or molecular sieve chromatography or simply gel filtration chromatography. As the name indicates, this type of chromatography will help us to include some solutes inside that or exclude some solutes inside it. Finally, we can get the solutes present in the mixture separated inside the column and we can elute those accordingly. In gel filtration chromatography, the porous polymeric matrix formed of spongy particles with pores completely filled with the liquid phase or mobile phase. The diameter of the pores is critical as separation is based on the molecule size. The interior of the pores is reached partially or wholly by smaller molecules. The larger molecules of the sample are excluded and so they will come out earlier. The gels or the polymers of the stationary phase consist of open three-dimensional networks formed by cross-linking of long polymeric chains. The pore size varies with the degree of cross-linking. Then, just like other gel chromatographic techniques, the sample is layered on the top of the column and then separation occurs. And finally, by the help of mobile phase or any kind of eluent, we can elute the different types of solutes present in the mixtures one by one. Next is hydrophobic interaction chromatography. The interaction between the hydrophobic molecules in an entropy-driven process based on the second law of thermodynamics is used for hydrophobic interaction chromatography. It explores the interaction between hydrophobic regions of proteins and hydrophobic ligands present on the chromatographic column. Proteins with higher hydrophobicity will bind more strongly to the ligands present in the stationary phase, while less hydrophobic proteins or more hydrophilic proteins will bind less strongly. So during elution, we will get the hydrophilic proteins first, then the medium proteins, and then finally we will get the hydrophobic proteins as our eluent. The next variety of chromatography is ion exchange chromatography. In ion exchange chromatography, the stationary phase contains one kind of charge, say for example, positive charge. In ion exchange chromatography, the gel matrix is a kind of resin which carries fixed charges, and these charges are not exchangeable. And this stationary phase contains mobile active ions which are called counter ions, which are loosely attached to the matrix. These loosely attached ions can be exchanged with the similar kind of ion present in the sample. For example, the fixed charges are positive, the mobile active ions or counter ions are negative. Now, when a sample is poured for separation process, all the solutes which contains negative ions and present in the sample, they will substitute the counter ions, that is the negative ions present as the counter ions in the stationary phase. Therefore, the negative ions attached as counter ions in the stationary phase, they will come out of the gel, and then all the solutes which carry positive charges in the sample, they will come out of the gel column. And finally, when we will add some kind of eluent which also contain negative ions and has greater affinity for binding with the positively charged ions present as fixed charges in the stationary phase, will come out. Therefore, first we will get all the counter ions which are negative ions out of the column. Then all the positively charged solutes, they will come out of the column. And finally, when we will add some eluent which contains negatively charged ions, they will help us to get the negatively charged solutes out of the column. Next is affinity chromatography. It uses biological interactions to separate and analyze some sample components. It's a powerful method for purifying specific molecules or groups of molecules from complex mixtures. A support medium such as cellulose beads is used as the stationary phase. A substrate or coenzyme is bound to support medium so that the reactive groups are exposed. Here in this image, you can see that the chromatographic support matrix contains the red flags, which may be antibodies, which may be some type of substrate or enzyme or coenzyme, etc. Now the sample will be added. Then all the sample molecules which can bind with a specific binding part of the fixed antibody or coenzyme or substrate, etc., they will be retained within the column, and everything else will come out of the column. Then when we will add some eluents which can replace the bound molecules, we can get extremely purified solutes out of the column. As the picture shows, the green molecules will come out of the column after elution. Affinity chromatography is highly specific, so we can get highly purified molecules from this. So that's all for today. If you have any question, you can write those in the comments. Till my next video, stay healthy, stay safe, stay tuned. Bye.